Just wanted to take this opportunity again to thank the organizers for inviting me for a really excellent 2 days of discussion. And really, it's good to know that you have the same issues that to deal with that we do as well. And we have a lot of things in common in terms of unanswered questions in the management of our patients. So I just wanted to give you a foray into where we think the future of immunosuppressive therapy is going, what we might be seeing down the line, and also give you an overview of the sort of things that we're currently doing in terms of managing things like antibody-mediated rejection, where, as Dr. Ottawa mentioned, therapy is much less well defined. And this is data from our group before we moved from UCLA to Cedars-Sinai, showing the rates of what we call cellular rejection and non-cellular rejection, which is probably AMR, down to 2010, where with progressive initiation of the newer therapies, we see that the rates were pretty low. And right now, our cellular rejection rate's probably in the 5-6% range. But our AMR rates have actually started going up simply because we're doing higher risk patients. And we know that MMF is superior to azathioprine due to with improved survival, as was shown in this randomized trial. And in the 3-arm study, the combination of TAC and MMF was far superior to cyclosporin and MMF, and TAC and sirolimus may be a little bit better than TAC and MMF. And certainly, we all know that we struggle with issues of rejection and infection within the first year post-transplant. But in the long term, what really limits the survival of organs is the development of chronic rejection or allograft vasculopathy. And more commonly, as we transplant older and older patients, we're seeing malignancy as being a significant problem, limiting survival. And I just wanted to kind of reiterate the issues relating to AMR. This is a consensus conference that we held a few years back, and we did a survey prior to the conference to try and get a global picture of how common AMR is. And essentially, 46 centers reported about a 6% incidence of treated AMR. And the total number of AMR patients with a PRA greater than 10% before transplant was about a third. And the total number of sensitized patients treated pre-transplant to reduce their antibodies was also about a third. The other characteristics of AMR included the observation that 70% of treated AMR patients recovered with an ejection fraction greater than 45%. But 20% of AMR patients had developed recurrent AMR. So it is a problem that kind of tends to linger on. I'm not going to belabor this point, but Dr. Ottawa gave a nice review of the various therapeutic options for AMR that are currently used, but I will kind of delve into the mechanisms of some of the agents that have used. IVIG is probably a very common agent used for the management of both antibodies and antibody-mediated rejection, and really the benefit occurs due to its pleiotropic effects on on antibodies, and this includes its effects from the FAB portion of the molecule, resulting in ADCC and cell depletion, blocking cellular receptors, abrogating cytokine and autobody neutralization, and also scavenging anaphylotoxins. And if you look at the FC portion of the molecule, it's beneficial in saturating FC receptors. It has a role in expanding regulatory T cells, blocking activating receptors, modulating dendritic cells, and also modulating FC receptor expression within plasma cells as well. So a number of different effects with IVIG. Rituximab is a humanized monoclonal antibody against CD20, which essentially knocks out a large line of B cells, from pre-B cells to even memory B cells, but has no effect on plasma cells, and it's the plasma cells that make the antibody. But it is a useful drug in preventing the maturation of B cells to the plasma cells. And I mentioned yesterday our protocol that combines the use of IVIG and rituximab in management of circulating antibodies, and this protocol has also been used in management of antibody-mediated rejection. And this is our data from a few years back showing that it does work. It doesn't work in every patient, but in general you do see a decline in PRA levels when you use IVIG and rituximab, and some of these patients also received plasmapheresis as well. However, despite these therapies, they do have a higher risk of developing rejection post-transplant, although survival is not really affected post-transplantation. What about Bortezomib? This is a proteasome inhibitor against plasma cells. The role of the proteasome is to scavenge misfolded proteins within the cytoplasm so that immunoglobulin molecules can then be excluded from secreted from the cells. The accumulation of misfolded proteins within the cell can lead to apoptosis, and the proteasome plays an important role in being a scavenger for these misfolded proteins. What Bortezomib does is it inhibits the proteasome so that these misfolded proteins accumulate within the cells and induce apoptosis of plasma cells. And this is the protocol that we use involving plasmapheresis and Bortezomib. We use plasmapheresis in conjunction with Bortezomib because when you deplete immunoglobulins from the circulation, you actually upregulate plasma cells to making more immunoglobulin. And so when you make more immunoglobulin, you can then hit them with the proteasome inhibitor, and so that enhances the apoptotic effect of the treatment. So the combination works better overall. And this is our experience in 21 patients where the antibody does come down following therapy, and those antibodies actually stay down even post-transplant. So the effect is durable if patients respond to the therapy. And it's pretty well tolerated. The adverse events for this type of therapy generally is restricted to infection, and usually line infections are what we see. 8% gastrointestinal neuropathy and thrombocytopenia are the side effects from the Bortezomib. Thrombocytopenia is really the most common and noted in about 80%. And we work with our oncology colleagues to dose Bortezomib. At our institution, only the oncologist can prescribe this drug, so they monitor the parameters quite well for this. And if we look at the post-transplant outcomes for these patients, we do see an increased risk of rejection post-transplant simply because they're sensitized patients, but survival is really not affected and is comparable to non-sensitized patients. One drug that has been of particular interest for me has been eculizumab. Dr. Atwar mentioned its mechanism. It's a terminal complement inhibitor which prevents the formation of the membrane attack complex. It is available in the United States and approved for use in the treatment of paroxysmal nocturnal hemoglobinuria and atypical hemolytic syndrome. And the reason we were interested in this was because in the renal transplant literature, it was shown to have a significant impact in reducing AMR rates post-transplant. So we've been involved in the DUET pilot study where eculizumab has been administered to very highly sensitized patients whom we have accepted a donor and have gone across donor-specific antibodies. And these patients receive standard induction protocol, methylprednisolone, ATG for 5 days, followed by IVIG for 2 days. But in addition, they get eculizumab at release of cross-clamp and further dosing for the next 2 months. Standard maintenance immunosuppression consisted of tacrolimus, mycophenolate, and prednisone. And our preliminary outcomes, we now have completed our study with 20 patients and are awaiting 1-year follow-up on our last patient, which will be in July prior to submitting this for publication. We see that survival is actually very good at 88.5%. We had one patient who actually died in the OR at the time of transplant, had a total artificial heart, was aggressively treated for a fairly high rate of sensitization. Unfortunately, in the OR, she had pus around the entire device, and she didn't make it out of the OR. She received the drug, so she's included in the study. We had one other death of a patient dying about 6 months post-transplant from development of accelerated vasculopathy, likely from her antibody burden. But the remainder of the patients have actually done very well. There has been no cellular rejection. There has been more AMR noted on a biopsy, but that's expected with this drug because it's a terminal complement inhibitor, and so it doesn't prevent activation of the proximal components of complement. So you will see C4D on the biopsy. But none of these patients who are surviving have had any form of graft dysfunction, despite the fact that the majority of the patients have donor-specific antibodies. The other observation we made with this drug is that donor-specific antibodies actually decline post-transplant with this agent. And the therapy was actually pretty well tolerated, although half the patients had some form of infection, which were manageable. Another drug that we're about to embark upon a multi-center NIH-funded trial is tocilizumab, which is an interleukin-6 receptor antagonist. Interleukin-6 is an interesting cytokine because it plays a key role in shaping both T and B cell immunity. With regard to T cells, IL-6 plays a significant role in production of TGF-beta and IL-10, which has a role in tolerance and preventing autoimmunity. It has a role in the production of interferon gamma, a bunch of other interleukins as well, which are, have a role in allergy, atopic disease, and autoimmunity. And in fact, tocilizumab is now used for rheumatoid arthritis. It also has a critical role in B-cell differentiation to plasmablasts, and it is felt that it may be beneficial in preventing the development of antibodies. It's a first-in-class humanized monoclonal antibody directed against the IL-6 receptor. It's used for rheumatoid arthritis and juvenile arthritis, as well as graft versus host disease, and has shown to have impressive efficacy in rheumatoid patients who have failed to respond to first-line immunosuppressive agents. And this is data from our renal colleagues at Cedars-Sinai, Stan Jordan, who initially looked at the use of tocilizumab and IVIG in patients who really didn't respond to other forms of desensitization therapy. And that in the renal population, about a third of the patients really failed to respond to plasmapheresis, IVIG, or rituximab. And so they did this study in about 10 patients to see if, what the effect was. And what they found was that it was actually very effective in reducing donor-specific antibodies. These patients who were treated ended up with a significant decline in DSAs, and it was actually eliminated. DSAs were eliminated in all but one patient who had 2 weak DSAs at 12 months, but no antibody-mediated rejection on protocol biopsies. So based on this renal experience, we're embarking upon a clinical study for routine use as an induction agent at the time of a transplant. This will be a randomized study to see how effective this is going to be. And this is just a data on looking at allograft survival rates in the tocilizumab patients compared to standard of care patients in that renal transplant population. Small numbers, but very interesting data. So just to summarize, we know that advent of cyclosporin and azathioprine facilitated heart transplantation with improved outcomes. Advances in maintenance immunosuppression with tacrolimus and MMF reduced cellular rejection rates significantly. However, the number of sensitized patients awaiting transplant is increasing, and despite the use of a virtual cross-match, AMR for our population remains the issue that we really are struggling with. Newer immunosuppressive therapies targeting B cells continue to evolve, but require really robust clinical trials. I didn't talk about proliferation signal inhibitors too much, but for maintenance therapy, there may be incremental benefits, but in our population at least, they seem to be fairly poorly tolerated. About 30% of patients don't tolerate PSIs. It's interesting, in Europe, the tolerability is much greater. The Germans are able to get their patients on much higher doses, and I don't know what the experience is here in terms of tolerability of these agents. The newer targeted therapies offer promise, but really require ongoing clinical evaluation. Thanks for your attention, and again, thank you for inviting me to this great conference.